Welcome back, guys. Now in this video, let's discuss about transport of substances across cell membrane. So what are the different types of transports? Here, let's begin with the basics. If energy is required in the transport of the substances, then it is called as active transport. So now what is the first type of transport? Transport is mainly divided into two types. What are they? First one is called as active type of transport as well as passive. Okay, active transports and passive transports. So what is the difference between active transport and passive transport? Difference is that if ATP is involved, okay, if ATP is utilized, energy is utilized in the process of transport of the substances, then it is called as an active transport. Okay, ATP, or I should say actually energy. Okay, energy is involved. In passive transport, no energy. So no energy or ATP is involved. Having said that, first let's begin with our topic, active transport. You already know in active transport, energy is involved. In active transport, you should know the substances are getting transported and energy is involved. But why energy is involved? Why we need energy to transport the substances is because substances are transported from low concentration to high concentration. Okay, low concentration to high concentration. The substances are moving from low concentration to high concentration. Normally, we know if anything is moving from a high concentration to low concentration, there is no need of any energy. By nature, it will move. From high concentration to low concentration, substances move. But now, if any substance, if you want to move a substance from low to high concentration, that is against concentration gradient. Normal concentration gradient is what? Concentration gradient is the difference between the concentration. Usually substances move according to the concentration gradient, that is from high concentration to low concentration. But if you want to move a substance from low concentration to high concentration, you need to give energy. Okay, simply try to understand like this. For example, this pencil, okay, this pencil, if you leave this, it will come from high to low. There is no need of any energy, it will simply fall. But if I want to throw this pencil up, I need to give energy. Why? Because I am doing against the gravity. Okay, so I have to give energy to move this pencil up, something like that. Okay, now active transport, energy is required. Okay, how many types of active transport are there? There are two types. Okay, there are two types of active transport. What are they? The first type is called as primary active transport. Second type is called as a secondary active transport. So what is the difference between primary active transport and secondary active transport? Remember, in both primary as well as secondary, energy is involved. Substances are moving from low concentration to high concentration. That is against the concentration gradient. But what's the difference, sir? Is in primary active transport, there is direct use of energy or direct use of ATP. ATP is directly used at the site of transport. In secondary active transport, energy is used indirectly. Indirect use of ATP. ATP is utilized, but not at the primary site, not at the site of transport, but somewhere else. Okay, I will explain you in detail what is primary active transport, secondary active transport, but just for now remember primary active transport, direct use of energy, a direct use of ATP. In secondary active transport there is indirect use of ATP. Okay, now after this let's begin with the primary active transport now. What exactly is this primary active transport? Okay, first let me write here primary active transport. Okay, what are the examples of primary active transport? You know, direct use of energy, direct use of ATP. What are the examples? If you ask me examples of primary active transport, the best example, number one example is sodium, potassium, ATPase. Okay, sodium, potassium, ATPase. Second example is calcium, ATPase or 𝐒𝐻𝐫𝐞. Okay, sarcoplasmic, endoplasmic reticulum, calcium, ATPase. Okay, H plus proton, ATPase. So these are some examples. But one thing you can identify, appreciate, see anything which ends with the word ATPase. Okay, so anything which ends with the word ATPase, all of them they are examples. All these transporters, these are the transporters, sodium, potassium, ATPase, calcium, ATPase, proton, ATPase. Wherever you see these transporters which are ending with the name ATPase, all of them they are examples of primary active transport. Okay, all of them are examples of the primary active transport. For example, let me start with the sodium, potassium, ATPase only, the most important one. You look here, imagine this is one cell. Okay, example number one I am giving here. This is a cell. Now tell me, in a cell, okay, inside the cell, which ions are more in concentration? We know cells are bags of potassium. Inside the cell, the potassium concentration is more. Okay, outside the cell, who is more? Outside the cell, more sodium concentration is there. Sodium is more outside the cell, potassium is there more inside the cell. Okay, now see, there is this one transporter. Look here, there is this one transporter. There is this protein. What this protein it will do is, it will take sodium which is present inside the cell. Even sodium is there inside the cell, but less quantity. Okay, less sodium is there inside the cell. Okay, for example, say only very few sodium is there, but more sodium is there outside the cell. Okay, so this transporter, what it is doing is, it is taking this sodium out of the cell. It will take 3 sodium ions out of the cell, and same transporter it will try to bring 2 potassium ions into the cell. Okay, 2 potassium are coming into the cell in an exchange for 3 sodium. But the problem here is what? See, already outside the cell there is more sodium. More and more sodium is there outside the cell. What is the normal concentration gradient? Sodium can move from out to in, from high concentration to low concentration. Normally sodium have to move from out to in, but it cannot cross. Why? Because it's a positive thing, right? It's a positive charge. Normally charged things cannot cross the cell membrane. Okay, but there is this one transporter. What it is doing is, it is taking the sodium from the cell and it is pushing out of the cell. So 3 sodium are going out of the cell in an exchange for 2 potassium. Potassium also, see, already potassium concentration inside the cell is more, but still potassium which is less outside, potassium is now moving from low concentration to high concentration. Potassium concentration outside the cell is less potassium is there, but still the potassium which is low outside is coming into the cell, so low to high concentration. So do we need energy or not? We are moving the substances against the concentration gradient. The sodium, potassium, ATPase, this pump is called as, now let me write here, the name of this pump here is called as sodium, potassium, ATPase. This sodium, potassium, ATPase, okay, it is using ATP. Now it will use ATP, energy, to move the substances across the cell membrane against the concentration gradient. Okay, so wherever you see this word ATPases, sodium, potassium, ATPase, example of primary active transport. The sodium, potassium, ATPase are present on almost all the cells. It's a universal transporter. It's present on all the cells. It brings 2 potassium into the cell in exchange for 3 sodium. Okay. Now if you ask me what is the function of the sodium potassium ATPase, see the sodium potassium ATPase, it plays a role in the maintenance of the resting membrane potential. It maintains the voltage of the cell. Okay, by sending 3 sodium out of the cell and bringing 2 potassium into the cell, it maintains the resting membrane potential of the cells. Okay. And not only this, later on I will tell you, the sodium potassium ATPase is the one which provides energy, indirect energy, provides. Okay, just remember this point now, later you will get more clarity. Okay, provides energy for secondary active transport, provides energy for the secondary active transport. Okay, the sodium potassium ATPase by itself, it is an example of primary active transport. It gives energy for the secondary active transport. How? Later I will explain. Next, you will have a doubt, sir, what is this 𝐶𝐶? Okay, and what is this calcium ATPase? Where it is present? See, this is your skeletal muscle. Okay, this is the cell membrane of the skeletal muscle. Now, please tell me, this is called as sarcolemma. Okay, sarcolemma. See, now it is invaginated. It's having this invaginations. Now, you know these invaginations, what are they are called as? They are called as the T-tubules. You already know it. Okay, T-tubules. And inside a skeletal muscle, there is one more tubule present. What is this one more tubule called as? See, these tubules, they are present longitudinally. See, these tubules are called as the L-tubules. Okay, this is L-tubule. See, this L-tubule is nothing but sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. Now, look here, inside the sarcoplasmic reticulum, this is a very, very important MCQ. Inside the sarcoplasmic reticulum, what is present? Calcium is present. So, sarcoplasmic reticulum, it's a store of calcium. Normally, calcium, it is not there in the cytosol, it's not there in the sarcoplasm. Calcium levels inside the cell is very, very low. Where calcium is present? Calcium is present inside the sarcoplasmic reticulum. It's not freely available. Okay, so calcium is there. Now, during muscle contraction, what happens is, via the ryanodine receptors, there are channels called as the ryanodine receptors, which you will study in detail in the in the muscle physiology, not smooth muscle. In the muscle physiology, we will study about what is this dihydropyridine receptors, ryanodine receptors, what are they? You will study in detail. But what I am telling is, when the muscle is getting depolarized, okay, the alpha motor neuron is giving stimulation to the muscle. Muscle is depolarized. The muscle membrane is depolarized. When the muscle is getting depolarized, what happens? It have to undergo contraction. First, depolarization should be followed with the contraction. Now, when the muscle is depolarized, see, the calcium, it will come into the cell. Okay, calcium is leaked into the sarcoplasm. Now, this calcium, what it will do? It will go and help in the muscle contraction. Okay, it will go and help in the muscle contraction. So, contraction completed. Do you want a sustained contraction? No, we don't want that. Contraction should be there, and later you have to have the relaxation. Contraction should be followed by relaxation. So, contraction, calcium is helping. After contraction, what should happen to this calcium? What should happen? This calcium should again have to go back into the sarcoplasmic reticulum. It have to again go back. I have already taught you, inside the cell, here the calcium concentration is less. This calcium, whatever is released, yeah, little amount of calcium is released. But inside the sarcoplasmic reticulum, what about the calcium concentration? The calcium concentration is super high. So now we have to move this thing, this calcium, we have to move this calcium from low concentration, that is in the sarcoplasm. Now the calcium is in the sarcoplasm. In the sarcoplasm or cytoplasm, they are the same. Okay, sarcoplasm is the cytoplasm of the muscle fibers. So now this calcium have to move from low concentration into the high concentration. They have to go back into the sarcoplasm. So there is this one pump present here. So what is the name of this pump? This name of this pump is called as 𝐶𝐶. Okay. So what is 𝐶𝐶, sir? S stands for sarcoplasmic endoplasmic reticulum. Okay, sarcoplasmic endoplasmic reticulum. Ca for calcium ATPase. So 𝐶𝐶 means sarcoplasmic endoplasmic reticulum, ATPase. Okay, sarcoplasmic endoplasmic reticulum, ATPase. So now tell me what it is doing. It is allowing the calcium from low concentration to high concentration. Okay, so calcium is getting back from low concentration, again it's going back into the cell, it's getting concentrated. So that's why as a substance now here in this condition, as a substance is moving from low concentration back to high concentration. So this is an example of primary active transport. Okay, so 𝐒𝐻𝐫 is an example of primary active transport. The 𝐒𝐻𝐫 is nothing but a calcium ATPase. It's a calcium pump. It's a calcium pump. So the second example is also completed. This is the first example, sodium potassium ATPase. Second example, calcium ATPase. And even this proton pump, it is present in parietal cells. It's present in the parietal cells of gastric mucosa. See, in the gastric mucosa, in the stomach mucosa, there are depressions called as the gastric glands. In these gastric glands, there are cells called as parietal cells. These parietal cells, what they are doing? These parietal cells, on the surface of these parietal cells, they have proton ATPase. They have the proton ATPase. What this proton ATPase, what it will do is, this will move the protons into the lumen. It will move the protons into the lumen. So the protons will bind with the chloride and forms the HCl. Okay. So proton ATPase, and in simple way you can say proton ATPase are present on the parietal cells. Parietal cells are present in the gastric mucosa. So these parietal cells are continuously producing these protons. Protons are nothing but the acids. So parietal cells are the one which produces the acid in the stomach. Okay. Just remember that. Enough. Now, after this, I need to add a few more points regarding the sodium potassium ATPase. Sodium potassium ATPase is an example of primary active transport. Okay. See, look here. This is the cell membrane. Imagine this is the cell membrane. Okay. This is outside the cell. This is inside. Outside and inside. Now, in this bilipid membrane, there is this transporter present. Okay. Which is called as sodium potassium ATPase. The sodium potassium ATPase, it is having alpha subunits and beta subunits. Okay, here for easy understanding purpose, I am showing you this as alpha subunit and this as beta subunit. Now, normally it's not like that. There are two alpha subunits, two beta subunits will be there, like, but it's made up of alpha subunits and beta subunits. See, this alpha subunit is considered as regulatory unit. Alpha subunit is regulatory in nature. Alpha subunit is present even outside as well as inside. So, inside the cell, to this alpha subunit, intracellular, to the intracellular domain, who is going to bind? Say ATP as well as sodium. Okay, so ATP and sodium, these molecules are going to bind to the intracellular side. They will bind to the intracellular side and from there they will go out. Okay, and on the extracellular side, who will bind? Potassium is going to come and bind on the extracellular side. So ATP binding site is present on alpha subunit or beta subunit? It's present on the alpha subunit. Intracellularly or extracellularly? Intracellularly. Potassium is going to bind on extracellular side. Okay. Next. So you know about the alpha subunit, beta subunit. See, this beta subunit, it is having the glycosylation site. There are 3 glycosylation sites. So beta subunit have sites for glycosylation. Now already I have taught you, in alpha subunit, who are going to bind? Sodium and potassium, sodium as well as ATP binds intracellularly and potassium binds on extracellular side. Okay, potassium binds on extracellular side. What are the other important points which you should know? The sodium potassium ATPase, what it is doing? It is bringing 2 potassium ions into the cell and throwing 3 sodium out of the cell. Okay, 3 sodium ions are going out of the cell and bringing 2 potassium ions into the cell. So what is the ratio? The ratio is called as, first this ratio is called as the coupling ratio. Okay, coupling ratio. So what is the coupling ratio? It's 3:2. 3 sodium ions are being exchanged for 2 potassium ions. So this is the coupling ratio. Okay. Next, the sodium potassium ATPases. Sodium potassium ATPase activity. The activity of the sodium potassium ATPase is regulated by whom? See, there are certain hormones which will activate the sodium potassium ATPases, and there are certain hormones and substances which will inactivate the sodium potassium ATPases. Now, first let me write here, the number 1 hormone is thyroid hormone. The thyroid hormones what they will do? They will activate the sodium potassium ATPase. They will activate the sodium potassium ATPases. They will increase the number of sodium potassium ATPases. And not only this, there are drugs like digitalis. Okay, digitalis group of drugs like digoxin, digitoxin. This is digitalis, obeying, dopamine. See these drugs. Okay, whatever I have written over here, they will activate. Okay, they will activate. Sorry, they will inhibit, not activate. They will inhibit the activity of sodium potassium ATPase. But these thyroid hormones, they will activate. Okay, they will activate the activity of the sodium potassium ATPases. For your understanding purpose, I will tell you. See, look here. What is this gland? This gland is a thyroid gland. The thyroid gland. Inside the thyroid gland, we know there are these cells called as the follicular cells. Now these follicular cells, okay, they are all sitting just like a round, and what they are producing? So in the colloid, they are producing the thyroid hormone, and they are storing the thyroid hormones. So T3 and T4, the thyroid hormones are coming from the thyroid gland. Now these thyroid hormones, see, where they will come and act? Now they are coming and acting on the cell. Okay, they were acting on this cell. The thyroid hormones, they can directly cross the cell membrane. This thyroid hormone is crossing the cell membrane. And inside the cell, what do we have? Inside the cell, there is nucleus present. Okay. Now look at this cell. On the cell surface, generally, on this cell surface, there is this sodium potassium ATPase present. Okay. How many sodium potassium ATPases are present? For example, on this cell, 2 sodium potassium ATPases are present. But the moment thyroid hormones comes into the cell, see the thyroid hormone T3, the active form, T3 is coming into the cell. Now this T3, it will also cross the nuclear membrane. T3 is crossing the nuclear membrane. Now this thyroid hormone, it will bind with its receptor. So now tell me where the thyroid hormone receptor is present. The thyroid hormone receptor is present inside the nucleus. So thyroid hormones have intra-nuclear receptors. Okay, they have the intranuclear receptors. Okay, they are coming into the nucleus and binding with the nucleus. What they will do? Now this complex is going to act on the DNA, will cause now transcription. Now there will be transcription. There will be formation of the mRNA. Now this mRNA, it will come into the cytoplasm. In the cytoplasm, with the help of ribosomes, what happens? Translation will happen. So now the thyroid hormones are activating those genes which will form a protein. Okay, now certain genes are activated. Now transcription and translation is going to happen. After translation, do you know what happens? Which protein is going to form? Many proteins, but the most important protein which I am discussing here is, look here, the thyroid hormones, whenever they are acting on the cell, see more number of sodium potassium ATPases. So more number of sodium potassium ATPases are coming out of the cell. So whenever more sodium potassium ATPases are there, see thyroid hormones are activating the sodium potassium ATPases, and the number of sodium potassium ATPases are also increasing. So now more number of sodium potassium ATPases will break down more number of ATP. They will break down more number of ATP. So more energy is being broken down, more ATP is getting utilized by the cell. More ATP is getting utilized by the cell. More number of calories are utilized by the cell. Okay, in the transport of sodium and potassium. So what happened to the basal metabolic rate? So basal metabolic rate increases. Okay, this is the very important question. See, I have little deviated from the topic, from the transport, because you should know the sodium potassium ATPase, which is an example of primary active transport. Okay, the sodium potassium ATPase activity is under the control of which hormone? Thyroid hormone. When thyroid hormone is acting on the cell, what happens? Thyroid hormone, it will bind with its receptor in the nucleus, causes the transcription and translation, so that at the end of the day, the more number of sodium potassium ATPase are getting produced. Now, because more activity is there, more sodium potassium ATPase, more ATP are getting broken down, because of this, the basal metabolic rate in the body increases. So, this is the reason why we say thyroid hormones increases the basal metabolic rate. How? By increasing the number of sodium potassium ATPases. Sodium potassium ATPases are doing what kind of transport? Primary active transport. Okay, primary active transport. So, okay, done. Next, after this, let's discuss about secondary active transport. Secondary active transport. So, what exactly is the secondary active transport? Energy is utilized or not? Yes, energy is utilized. Okay, energy is utilized. Energy is involved in this process. Energy is present, positive, but indirect. So, what do I mean by indirect? Okay, indirect usage of energy. First of all, let me tell you what are the examples of the secondary active transporters. So, I will explain that example so that you will have a better clarity. Examples are secondary active transport: sodium, glucose, co-transporter. Okay, sodium glucose co-transporters. Their example of secondary active transport are sodium, potassium, 2-chloride Symporter is an example, or sodium chloride Symporter. So these are the examples of secondary active transport. Now first, the most important one every time which comes in your exam is this sodium glucose co-transporters. How this is an example of secondary active transport? Look here. Now imagine this is a nephron. So this is the Bowman's capsule. Here is the Bowman's capsule. Down to the Bowman's capsule, what do we have? Down to the Bowman's capsule, we have the proximal convoluted tubule. So this is the proximal convoluted tubule. Okay. Now look here. This proximal convoluted tubule, it is going to continue as loop of Henle, descending limb of loop of Henle, hairpin turn, ascending limb of Henle, you know it. Now here I am talking about this cell, the cell which is present in the proximal convoluted tubule, PCT. So there is this cell which is present in the proximal convoluted tubule. Now look here, I am showing you a capillary, okay, in the glomerulus. This is the glomerular area. Now this capillary is called as glomerular capillary. Okay, this is the glomerular capillary. Now see, normally here's who is coming? Sodium is coming. Blood, like just like plasma is passing, even in the plasma sodium is there, glucose is there, amino acids are there, so many things are there. Now sodium, will it be filtered or not into the nephrons? Yeah, sodium is filtered. Glucose, will it be filtered or not? Yes, glucose is filtered. Sodium, glucose, everything is getting filtered. Most of the students will think, glucose is not filtered in the kidneys. Glucose is filtered, but do you think glucose will be lost in the urine? No, glucose is not lost in the urine. Glucose will be reabsorbed back. 100% of the glucose will be reabsorbed. No glucose molecule is lost in the urine. Okay. So, see, the sodium is coming like this, and glucose is also coming like this. Point here which I want you to know is, see, the glucose concentration inside this PCT epithelial cell, this glucose concentration in this cell is very high. These cells have high amount of glucose. Normally, these PCT epithelial cells, they have a capacity for gluconeogenesis also. So, lots and lots of glucose is already there here. But what about the glucose concentration in the tubule? In the tubule, glucose concentration is low. Okay, glucose concentration is less. In both the places, in the tubule, in the urine, glucose is there, and in the cell also glucose is there. But the glucose concentration in the cell is more, but the glucose concentration in the tubule is less. Now, let's talk about the sodium. Okay, let's talk about the sodium. The sodium concentration inside the cell, is less. Okay, less sodium is there inside the cell. But the sodium concentration inside the tubule, when compared to the cell, it is more. That's why I am showing you. Please appreciate the sizes. Sodium in the tubule is big, means more sodium is there in the urine and less glucose is there in the urine. More glucose is there inside the cell, but less sodium. Now, tell me, which substance can move, which substance cannot move? See, there is a concentration gradient. There is a concentration gradient for the sodium. Sodium can move from high concentration to low concentration. So, sodium can be easily reabsorbed, no problem. So, sodium can simply reabsorb like this. Why? Because from high to low, no problem. But if you want to reabsorb this glucose, then it's a problem. Okay? Why? Glucose cannot move from low concentration to high concentration. There is a problem. You need to give the energy. So, how this reabsorption happens? Now, I will explain you. First, question number 1. Why the sodium inside the cell is less? Why it is less? It is less because of this transporter. See, there is this transporter here. What is the name of this transporter? See, this transporter, you should tell me. This transporter is throwing 3 sodium out of the cell. 3 sodium ions out of the cell. And the same transporter is bringing 2 potassium ions into the cell. So, 2 potassium are coming into the cell and 3 sodium is going out of the cell. Okay. So, continuously sodium is going out of the cell by using the ATPase. Energy is involved here. You know it. This is the sodium potassium ATPase, no doubt. This is the sodium potassium ATPase. By using the energy, sodium is kept low inside. Okay, by using the energy, sodium is now throwing out of the cell. So, intracellular sodium levels will go down. Now, see what happens. See, there is a transporter here. Okay, let me show you with the green color. Now, see, on the proximal convoluted tubule, there is this transporter present. Now, what this transporter is doing is, it will allow the sodium to enter into the cell. Okay, sodium is coming into the cell, no problem. Why? Because it's coming from high concentration to low concentration. When it is coming from high concentration to low concentration, it's coming according to the concentration gradient, from high to low. So, while it is moving in, there is no problem for the sodium. While it is moving in, it will take this glucose. So, while the sodium is coming into the cell, glucose will move from low concentration to high concentration. Okay. So, there is something called as, when a substance is moving from high concentration to low concentration, it will releases the energy. By using this energy, when the sodium is moving from high concentration to low concentration, it releases the energy. By using this energy, even glucose will come into the cell. So, this type of transport is called as sodium glucose co-transport. They are moving in one single direction and they are going together. So, sodium glucose co-transport. This is an example of secondary active transport. Okay, sodium glucose co-transport. Okay, is an example of secondary active transport. Why we are calling it as secondary active transport? See, number 1, sodium. It is going into the cell or not? Yes, it is going into the cell, sir. Glucose. Is it going into the cell? Yes, sir, it is going into the cell. So, transport of the substances is happening. Okay. But energy is involved or not? Energy is involved. Where energy involved? Here, this is the number 1 point where energy is involved. Here, ATP is utilized so that intracellular sodium concentration is kept low. As the intracellular sodium concentration is low, that's why sodium is entering into the cell. Okay. While sodium is entering into the cell, it also takes glucose into the cell. Okay. So, sodium and glucose are getting transported into the cell. but energy is utilized here. So that's why in the beginning I have asked you to remember, so the sodium potassium ATPases, what they will do? They will provide energy, they will give energy indirectly to the secondary active transport. If you look here, let me show you. See? Yeah, see here. So sodium potassium ATPases, they provides energy for secondary active transport indirectly. Okay. Now, what else I want you to know here is the name of this transporter. This transporter, what is it? This is called as SGLT. SGLT means what? S stands for sodium, GL stands for glucose. Sodium glucose co-transporter, T for transport. Sodium glucose transporter. Now, this SGLT, the number of this transport is called as SGLT type 2. Okay. Now tell me SGLT type 2, where it is present? SGLT type 2, it is present in the proximal convoluted tubule. Okay, proximal convoluted tubule. Okay, SGLT type 2. What is the coupling ratio? The coupling ratio of this SGLT type 2 is also important. It is 1:1. It will take one sodium ion into the cell and one glucose molecule into the cell. So, coupling ratio is 1:2. If you want to know a few more important points, SGLT type 1. Where this SGLT type 1 is present? SGLT type 1 is present in the intestines. Okay, in the intestine, we need to have absorption. In the kidneys, we have reabsorption. In the intestines, we need to have the absorption. So, there also sodium glucose co-transport only. But the transporter is different. The transporter here is SGLT type 1 in the intestines. What is the coupling ratio here? The coupling ratio here in intestines is 2:1. Okay, 2 sodium ions are needed to absorb 1 glucose. Okay, coupling ratio is little different. So, with this, the active transport, the topic, the primary active transport and the secondary active transport are completed. The secondary active transport, I have given you a classical example. But wait, one more point which I want you to know is, let me integrate this. See, there are certain drugs which will come and inhibit this transporter, SGLT type 2 inhibitors. Okay, SGLT type 2 inhibitors. Okay, SGLT type 2 inhibitors. In which condition we will use this SGLT type 2 inhibitors? Think logically and tell me. If this transporter is inhibited because of an external agent, do you think sodium and glucose will be reabsorbed? No, the glucose will not be reabsorbed into the body. The glucose will be lost in the urine. All the glucose is going out in the urine. So, there will be glucoseuria. So, in which condition we will this? We want the glucose to go out of the body. In which condition we want to decrease the blood glucose levels? Diabetes mellitus. So, SGLT2 inhibitors, they are used in treatment of diabetes mellitus. Okay. So, what are those drugs? DAPA, dapagliflozin, and canagliflozin. Okay, so dapagliflozin, canagliflozin and empagliflozin, okay, there are different types. They are the SGLT2 blockers, SGLT2 inhibitors. Now, if someone asks you, tell me, dapagliflozin and canagliflozin, they will act on which part of the body? Kidneys. In kidneys where? Nephrons. Nephrons where? Proximal convoluted tubule. They will block which transporter? Sodium glucose co-transporter type 2. So, because of using these drugs, what will happen? Blood glucose levels decreases. Okay, there will be decrease in the blood glucose levels. Urinary glucose levels increases. So, when the urinary glucose levels, whenever there is more glucose present in the urine, by using this glucose, microorganisms can grow. So, the side effect of these drugs is going to be urinary tract infections. Okay, UTIs, urinary tract infections. Okay, so the sodium glucose transporter type 2 inhibitors, okay, SGLT type 2 inhibitors are used in a condition called as diabetes mellitus, but the side effect of them is going to be urinary tract infections. I will give you a note, see here, note. It's just like a mnemonic kind of thing. Wherever you see ATPase, those transporters are example of primary active transport. Okay, they are examples of primary active transport. Wherever you see symporters, Okay, wherever you see the word Symport are co-transporters. Okay, Symporters are co-transporters. They are all example of secondary active transport. For example, look here. See, sodium glucose co-transporter, sodium potassium 2-chloride Symporter, sodium chloride Symporter. So wherever you see the word co-transporters or Symporters, they are all examples of secondary active transport. So with this, active transport is completed. In the next video, we will concentrate on the passive transport like simple diffusion, facilitated diffusion. Okay. Also, we will discuss about the vesicular transport, that in the next video. Hope the video is helpful. Thank you.